We're gonna just finish up by doing the reproductive system here. First, though, we'll do our classic thing and look at your very last warm-up and see what happened there. Let's go to the warm-up. Here we go. Okay. So which of the following glands release hormones that involved with regulating calcium? So this one you had to kind of remember that the thyroid gland had the calcitonin, which helped absorb calcium and kind of store it away. And the parathyroid was the one about releasing calcium stores and bringing calcium, kind of free calcium, back up in your body. I'm not quite sure, but a number of people wrote anterior pituitary, which doesn't have anything specifically about calcium. This one, name specific hormones. So tropic hormones, hormones that control the release of other ones. We looked at a bunch of these. They were actually all from the anterior pituitary. There was thyroid stimulating hormone, which talks to the thyroid gland to release thyroid hormone, adrenocorticotropic hormone, which talks to the adrenal cortex to release cortisol. Actually, you could also talk about the releasing hormones from the hypothalamus, like gonadotropin releasing hormone tells the pituitary to release LH and FSH. There are other releasing hormones as well. I didn't give them specific names, but I kind of talked about, like there's thyrotropic releasing hormone, which actually tells the pituitary to release thyroid stimulating hormone and things like that. So anyway, yes, make sure, this is kind of reminding me, just as a kind of public service announcement before your final exam again, make sure you read the questions carefully. One person like answered this question with just the name of a gland. It's like, name a specific hormone. So make sure when you're answering questions that you always kind of make sure you're answering the actual question, because it's easy to kind of get caught up in the moment. And this was just the big kind of adrenal cortex. Okay, calcitonin from the thyroid gland, epinephrine, most people got the adrenal medulla. Insulin, everybody got the pancreas. The other thing from the pancreas would be glucagon, which also everybody got. Oxytocin, everyone got from posterior. Growth hormone, everyone got. And so most people got most of these, this is good. Again, like I mentioned, on your exam, you should know the main glands, the main hormones released by those glands, and to a first order approximation, the function of those hormones that are being released. Any one of these topics can go into way amount of depth, but you should at least know kind of the basics. This one, all that drew about steroid hormones. So the core of it is their lipids. Remember, they're derived from cholesterol. Once you know it's a lipid, then you know that it's not going to dissolve easily in the plasma. That was the whole point about fats don't dissolve in water without help. So they're the ones that need carriers to travel in the bloodstream. They're most definitely not carbs. And again, most people got this. They bind receptors in the cell nucleus. They can actually drift through the membranes and end up into the nucleus, where there's, like I mentioned, there's these weird receptors that we don't have time to go into. So before we move on into the reproductive system, is there any questions or points of clarifications about endocrine that we've covered? All right. All right. And again, we actually introduced some of the main control of the reproductive system when we were talking about luteinizing hormone and follicle stimulating hormone, and talking about the follicle development in the ovary briefly. We're going to look at all that in more detail today. But we should start with, you know, what is the function of the reproductive system? It's a little more than you might first say. Oh, come on. Function of the reproductive system.
Create an offspring?
Yeah, create offspring. Keep the species going, right? But there are many different ways to go about that, right? If I was a sea anemone, I would just start kind of creating a little clone of myself. It would be a little person that looked just like me starting to grow off of my shoulder until it was big enough, and then it would break loose and be an independent free living organism. But that would be a total clone of me, the same genetic material and genetic code. What do we gain? This is really kind of wasteful. Most of the little sperm and eggs that get created never join up to become a new little human being. And there's this big, complicated social dance just to get the sperm and the eggs together in the first place. Why go through all of this kind of complicated process when other animals just make little clones of themselves?
Genetic diversity, so you can
Yeah.
survive many things.
So this idea of genetic diversity, it's worth, I think, just mentioning explicitly, because we think about some of the more kind of physical phenotypes, easily, like, the color of your skin or the size of your nose or your height or this or that. So there's some things that you can see that are pretty just obvious. But there's a lot of things about genetic diversity that are not obvious at all. We talk about genetic polymorphism. Meaning that there are different alleles for the same gene. Like, we've been like, if we go back to the nervous system, when we talked about clearing a synapse in the nervous system, we talked about, there's reuptake, which meant you had these little reuptake pumps that would suck serotonin back up into your presynaptic terminal, or sometimes there were enzymes that would break apart the neurotransmitters to clear the synapses. It turns out there are different versions of those enzymes. Some of them work faster, some of them work slower. There are different versions of those little reuptake pumps. Some of them pump faster, some of them pump slower. And it's not like there's one normal version and then there's some outliers. It's like a third of us have this version, a third of us have that version, a third of us have some other version. And that exists for all these different aspects of our nervous system, the little molecules that are running all of our wiring. It exists for a lot of the enzymes that are running all of our metabolic pathways. So even though the basic stories we've been telling, when you step back a little bit, are describing what's going on, kind of the details and the subtleties, there's a lot of differences when you go from person to person. There's this whole deck of different versions of different things, and they're all shuffled differently in each one of us. And it kind of gives robustness to the species, right? You've got COVID coming through, and some people end up on a respirator and are dead, and other people barely feel it, and keep going on and keep the species going to take over the planet. So this idea of genetic diversity really gives us robustness to the species. And it also is worth thinking about. You're all going, not, I don't know all, but most of you are going into healthcare, and you're going to just see all sorts of different responses to this same pathological processes, different responses to the same treatments. And it's worth remembering that, people all have their own kind of subtle differences that are going to have them respond differently to the same situation. So I think it's worth just kind of emphasizing, sexual reproduction means, you have, the parents mixing they have their own unique things, and their offspring is going to be some yet another unique mix of that genetic deck, and kind of keeps things going. Also, just, I know, but, there's lots, sexuality and gender and all that can get really complicated. Just for the purpose of our discussion today, we're just going to leave it as, female and male. It just makes life way easier, and it's going to let us just get into the core thing, because we're going to have females are going to make the ova. We're going to have the males make the sperm, the male gametes, female gametes, which are going to get together to make the new little human being. So we're just going to keep it at a simple level like that. So with that kind of introduction, we can kind of dive in. And when people talk about like primary sex organs, basically it's the gonads, which are going to be making the gametes. Right? Everything else is accessory. We are going to talk about, there's all the little tubes that carry the gametes to where they're going. There's, if you're in the woman, there's the uterus to help nurture and develop the little fetus. If it's in the guy, there's going to be all these glands to mix with the sperm to make the semen to, squirt out of the penis and all of that. But we all can, that's all going to be the accessory organs. The real core of sexual reproduction is going to be in the gonads, making the gametes. So let's talk a little bit about that process. Like, what is a gamete? What kind of cell division is actually responsible for making gametes? What specific kind of cell division makes gametes?
Meiosis.
It's going to be meiosis. So we need to talk a little bit about meiosis. And again, meiosis, it's only happening in the gonads, in the testes, in the ovaries. It's the only time you have meiosis going on. Everything else, once the sperm and the egg meet and become like a little zygote, then it's mitosis. That's all growth and development is mitosis. All of the repair of everything is mitosis. The only place you have meiosis is in the testes and the ovaries to make the gametes. But it's obviously a super important part of the process. So let's think about meiosis versus mitosis. I know you've all seen this in bio like 110, but it's worth, I think, reiterating the core differences because it's going to be important. They both start with some cell. And in a human, like one of your normal body cells is what we call 2N. Right? N is the number of chromosomes to define the basically the human blueprint. There's 23 chromosomes that make a human blueprint. And you got one copy from your maternal side, one copy from your paternal side. So you've got two versions of every gene, except for on the sex chromosomes, obviously, if you're a guy, because then you're X and Y. But basically that's your two alleles of every gene. You have one version from the mom, one version from the dad. So you have 2N, two copies of the genome. So that's where we start for both of these. And what happens next? How are meiosis and mitosis different in what happens as we start cell division? People got to remember this a little bit. I know you did it all with all little pipe cleaners and everything. Anybody?
Isn't there mixing of genes between the two chromosomes in meiosis?
Yeah, so meiosis is going to have a bunch of different things. One of the things, it's going to be mixing up those two sets of your chromosomes, the maternal and paternal versions. They're going to get shuffled and sliced and diced and reconstructed into unique chromosomes that have never existed before in the universe. And there's lots of other differences. Maybe I'll just, in the interest of time, I'll just kind of lay it out. Let's start with mitosis because it's kind of way simpler. Mitosis basically happens. You end up with two identical, basically clone daughter cells. You end up with two genetically identical 2N, which is also known as diploid daughter cells. Right, if you remember, there's every single chromosome will duplicate into the sister chromatids, and then they all line up along that metaphasal plate, and they all get pulled to each side during anaphase. So you end up with two identical copies of the original cell. Whatever this was, you get two identical copies here to move on for the most part. Meiosis is super different. Meiosis is going to have two. There's meiosis 1, and then each of those goes into meiosis 2. You're going to end up with 4 daughter cells. And if you remember during the metaphase 1, instead of just lining all the chromosomes along the that metaphase plate, you have the pairs, the homologous chromosomes are kind of sitting side by side, and only one or the other gets pulled to one or the other daughter cell. So we actually lose one of these ploidy levels. These are going to be 1N haploid daughter cells. These are each going to be 1N. And then there's also the genetic recombination, like this crossing over, where all of the the homologous chromosomes are sitting on top of each other and starting to swap in and out different parts of themselves. So you end up with these genetically unique. Each one of these daughter cells is haploid, and the genome that's in there is going to be some random mix of whatever your maternal and paternal chromosomes were. So this is kind of this weird mixing and matching of your two versions to create these unique, genetically unique haploid cells, right? And that's going to be the guy is doing this to make the sperm. The woman's doing this to make the eggs. And ultimately then you have a 1N sperm and a 1N egg. And when they get together, they become the 2N, the zygote. This 2N is going to be the unique genetic composition of the offspring, which is now a new 2N human being. And their paternal and maternal were whatever that sperm and the egg contributed there. Right? So this is going to be this constant mixing up of the gene pool here. So meiosis going on in the testes and in the ovaries. And we'll see, we're going to talk more specifically about spermatogenesis, the generation of the sperm, oogenesis, the generation of the eggs. They are both meiosis, although we'll see there are some specific differences as well in how it happens in the guy and in a woman. In fact, let's, for like spermatogenesis, obviously this is in the dude. Basically, you're going to have a cell. It's going to be called the spermatogonium. This is our little 2N. This is basically kind of like the stem cell that starts this whole thing. And it's going to undergo mitosis. And it's going to create a new cell that just replaces it, because it's going to we're going to want to keep going through this process again and again. The guy keeps making sperm from the moment he's gone through puberty till, it slows down, but he keeps making sperm his whole life. But the other daughter cell is going to commit into meiosis here. This is called a spermatocyte, primary spermatocyte. And I'm not going to get into the details. There's the secondary spermatocytes. There's the spermatids. Again, this is meiosis here. So this whole process we call spermatogenesis. And the final step, there is this crazy remodeling, right? You all know, everybody knows the cartoon of what a sperm looks like. A sperm does not look like a cell or a normal cell. A sperm looks like that. So there's this final step where most of the cytoplasm is thrown away. These little dudes grow a tail. That final step is called spermiogenesis. So spermiogenesis is that kind of remodeling. And we're going to see when we look at the testes in more detail, we're going to see there is these tight junctions that seal off the lumen of the tubes where these sperm are getting created. Because these are so different from your normal cells, you have to actually hide them from your immune system, or else you would start producing an immune response against them. They're very different from your normal looking cells. So this is all going on in the testes. Again, we're going to look at this in more detail in a few moments. But the resulting cells, these little sperm here, are very different. They have flagella. A flagellum is basically like a huge overgrown cilium. It's got this same microtubules. Remember the 9+2 arrangement of microtubules? It's got that same thing and wiggles, but instead of sweeping something across the surface of a cell, it's about propelling the entire cell through space. Lets it swim. It's got this little mid piece with lots of mitochondria because it's going to need a lot of energy to swim. It's got rid of almost all its cytoplasm. It's basically kind of like a nucleus delivery system, right? It's got the nucleus with the chromosomes, but not much else. And then the last part that it has is this kind of thing on its nose called the acrosome. The acrosome is going to contain enzymes that are going to allow it to actually get through this wall of cells that is surrounding the egg, which I might as well just show you this. So, if the sperm, this dinky little thing, the egg is huge. The egg is like one of the largest cells in a human body. It's like actually supposedly visible with the human eye. We're going to talk about this a little more later, but basically once the egg is ovulated and fertilized, it takes about a week of drifting through the fallopian tubes and getting down into the uterus before it starts actually hooking into the mom's nutrient supply. So it actually has to be completely self-sufficient for the first week of development. So this ovum has to be big enough to have all the nutrients and everything, all the supplies it needs to spend the first week of growth post-fertilization before it can hook into the mom's kind of nutrients. So it's really big. It's surrounded by cells. Remember, I mentioned last time that the ovum is never kind of by itself. It's always got these granulosa cells and stuff. Once the egg is ovulated, it still is surrounded by cells. These are the cells we call the corona radiata, which means kind of radiating crown. So it's just all these cells that are kind of surrounding the actual egg. So that acrosome has enzymes that help kind of digest and get through that. So as these little dudes all start coming in and they release these enzymes here, it'll start kind of chewing away at those cells there. So ultimately, one of these little dudes is going to be able to go, boom, make it in, puts its nucleus basically in to join with the DNA of the mom. Once that happens, there's all these things we call blocks to polyspermy. Once one sperm actually successfully fertilizes, there is all of these processes that happen. I'm not going to go into. There's basically this membrane that hydrates and actually just pushes off all the other dudes, and they're like dough. So yeah, once one gets in there, everyone else gets kind of pushed away. This thing that from this model, you can kind of see that the only thing you got from your dad is some DNA. Everything else, all of your cellular organelles, they're all from your mom's side. In fact, all of your mitochondria, which have their own unique DNA, it's all matrilineal. You can actually kind of follow matrilineal lines through mitochondrial DNA because it's only depends on who your mom was. It has nothing to do with who your dad was. Yeah, so the only thing you really get from your dad is the 23 chromosomes. Everything else was from your mom. So let's talk a little more about how this egg came to be, because that's going to be important as well. Let me spell that right. Oogenesis. So a few things I'll mention about oogenesis. First, it's meiosis, but because we want to end up with one big, huge cell when we're done, every time we do a cell division, one cell is going to get the lion's share of the cytoplasm, and the other one is going to basically get the booby prize. Not much, of just be a nucleus with maybe a little bit. And then the second cell division, this is not much there. This one will also divide. So there's only one viable egg, even though it's still the two divisions and with 4 potential offspring. Only one is going to end up being a viable egg because it's going to be always getting the lion's share of the cytoplasm and everything at each cell division. These other things that are left over, these are called polar bodies. The other thing is, when I talked about, remember I talked about spermatogonia, the kind of stems. In fact, let's go back here briefly. Whoops, not there. Here. In spermatogenesis, you had the spermatogonium, which was basically kind of the stem cell, and it divides, replaces itself, sends another daughter into meiosis to make sperm, but then this dude can just do it again and again and again. So this just goes on and on. In the female, the oogonium, the cells that ultimately create the eggs, they all commit into meiosis by the time you're born. So So all of the eggs, whenever you're going to ovulate and that follicle stimulating hormone starts going up and starts, getting one of the eggs to further develop, it's a little cell that's been sitting in suspended animation in your ovary since the day you were born. So basically these primary oocytes, and then it's got these little cells around it we call follicular cells. This whole thing is called a primordial follicle. And then if we look at a primordial follicle as this cell committed into meiosis but in suspended animation with these cells that are going to further develop to create the more elaborate structure around it, when you are born, this is like the ovary, you've got all of these little primordial follicles. Like 200,000 of them. So they are just kind of waiting there in suspended animation. It's part of, if you, there's that classic kind of ticking clock, like the longer you wait to get pregnant, the more of a chance there might be some, kind of genetic problems. It's because each of those cells has just been sitting there. It's not like they were just created when they were ovulated. They've been sitting there and waiting and waiting and waiting to finally continue the process. So these little follicles, again, the egg as well as the surrounding cells, once there is FSH, and this obviously starts once after menarche, when a woman starts reaching sexual maturity. So if FSH, this primordial follicle will start developing into a more complicated structure. And again, for this class, we don't need to really get into the details. But it's those little follicular cells start getting more complicated. They're called granulosa cells now. And then it gets even more complicated. And these fluid-filled spaces start getting developing in there until you get ultimately, I'm going to just draw the final step. So there's the egg. This whole big thing here, this is what we call the Graafian follicle. It's kind of ready to go. There's this fluid-filled space inside. Maybe I'll use my typer. It's called the antrum. This process of going from primordial follicle to this takes about 2 weeks. We're going to see when we look more in detail at the ovarian cycle and the larger cycle, this is kind of the midpoint. This is right before ovulation. And typically, when FSH is going up, there's more than one of these primordial follicles that starts developing. But as they go, as they keep developing, most of them kind of just die off. So maybe more, a few will start this process, but only one will successfully complete the process. The other ones just degenerate. I can give you the word. This official word is Atresia. Right, so a bunch of them start, but only one makes it all the way. Unless it is possible that you have more than one. And that's how you end up getting fraternal twins, right? If you have 2 eggs ovulated and they both get fertilized by different sperm, then you can actually have 2. It's basically like having siblings that were born on the same day. Which is different from identical twins, which we can talk about later. When people are taking drugs for fertility treatments, they often do on purpose, try to do multiple ovulations in order to increase the chance of a successful pregnancy. So FSH is going to create this. This follicle is also making estrogen, which is going to be important. Let me write that in here. So this structure is making estrogen, and it's kind of trippy. There's cells called thecal cells. Again, you don't need to know the details for our class here, but there's these cells more from the connective tissue called thecal cells that actually make testosterone. And then the granulosa cells are the ones that convert that testosterone into estrogen. And estrogen is there's actually not just, estrogen is a family of hormones. Beta estradiol is kind of the most important of them. So this structure is making estrogen, and we'll talk about it later, but the estrogen is going to be talking to the uterus and trying to get the lining ready to go for a potential pregnancy. So as the egg is developing in the ovary, the uterine lining, the endometrium, is going to be thickening and getting ready for a potential pregnancy as well. And so we'll be looking at that in more detail. What is the signal that triggers ovulation, triggers the egg getting ejected out? We talked about on Tuesday.
The luteinizing hormone?
Exactly. There's a spike in luteinizing hormone, and that causes basically this thing will, let me see if I can do this. Too much. Basically, the egg is going to go out and meet its destiny someplace. So it's going to hopefully get swept up by the fallopian tubes. We'll follow the egg a little later and see what happens if perhaps it meets a sperm. But what happens now is the remaining cells reconfigure and become the corpus luteum, which is going to be important for our story as well. So this becomes corpus luteum, and this produces progesterone. So I mentioned how the estrogen is talking to the uterine lining, getting it ready for a potential pregnancy. So now the egg has been released, and now the progesterone is saying, all right, we just sent an egg out. Hold your horses, keep all systems go. Because, sometime in the next week and a half, you might be getting some fertilized egg coming in ready to implant. So we want to make sure that the uterine lining stays ready and waiting. Spoiler alert, if there is no pregnancy, the corpus luteum will degenerate, the progesterone will drop, and in a couple of weeks, the uterine lining will slough off, and it'll be the menstrual flow if the progesterone is dropping. Right? So, if there is a pregnancy, we're going to see there's going to be another signal that keeps the corpus luteum going, keeps the progesterone up, keeps the That's why, a woman's going to miss her period if she's pregnant, because the progesterone stays high, which means there is never a hiccup and the uterine lining just stays intact, and there's no menstrual flow, no menses. So again, we're going to look at this in more detail, but just kind of setting the stage here, and to understand the whole cycle when we get to the female side. You got to remember, in the ovary, you've got these primordial follicles that are just lying in wait from the moment you're born. If the monthly cycle is going, the beginning, there'll be FSH, follicle stimulating hormone, which will stimulate one of these follicles in this 2-week process to become a mature follicle, which is making estrogen, which is preparing the uterus. This thing is going to ovulate at this, like around week 2. Egg goes off. The remainder of this follicle becomes this corpus luteum, which is now making progesterone, sustaining the endometrium in case there's a potential pregnancy. If there's no pregnancy, the corpus luteum degenerates, becomes this thing called the corpus albicans, and we'll see the process is basically going to start again. So we'll come back here. If you just take a look at the egg here, you can see as it's ovulated, it's still surrounded by some of these these granulosa cells that made up the whole follicle. That corona radiata that we were talking about, back over here, like that, is surrounding the egg. That's still those cells that were surrounding it when it was still part of the follicle, and that are still around it when it gets popped out into the fallopian tube. So, like, the egg is never on its own. It's always got like this little retinue of cells kind of holding it, taking care of it. So that is kind of that's actually kind of going into kind of more of the detail around spermatogenesis, which is kind of constantly happening, versus oogenesis, which, again, all the primordial follicles are committed into meiosis by the time you're born, and then post menarche, there's going to be some of those that continue on to develop into that mature Graafian follicle each month. All right, so now what we're going to do is we're going to go back into the testes, where the sperm are getting developed, where we have spermatogenesis going on. So let's look at the testes. And let's see how we're going to do this. Actually, maybe let's start with, let's draw like a guy. It's going to be a penis. There's going to be, so this is just supposed to be the abdominopelvic cavity here. The scrotum, usually people know what you're talking about if you put little pubic hairs on there. So, like, one of the big differences between the testes and the ovaries is that the ovaries stay up in the pelvic cavity, this nice kind of protected place. But the testes during development descend. There's a little tunnel. It's called the inguinal canal. It's a little tunnel through your abdominal wall. So the testes actually get lowered down. And end up in your this little, basically a little skin bag, here on the inside of the thigh. Why have the testes, which are at the, essential for the survival of the species, in such a vulnerable place, rather than keeping them up in the pelvic cavity where they're much safer?
Temperature control.
Temperature control. And why do we need to worry about temperature control?
I mean, if they get too hot, is there like a risk of damaging the sperm?
Right. So it turns out that this, for whatever reason, the sperm need to be a few degrees cooler than body temperature for normal production. So they have to be out where they can be cooler than body temperature. And there's a variety of things that help control the temperature. There is the cremaster muscle. Maybe I'll put that in here. The cremaster muscle basically attaches to the abdominal wall and can basically raise and lower the whole shebang in order to pull it closer to your body, to keep them warmer, let them dangle more if you want to give them a little more airflow. There's also another muscle within the skin, the dartos muscle, which can wrinkle or let smooth to increase or decrease the surface area of the scrotal skin. So you have a variety of ways to help adjust the temperature. Cremaster muscle is also going to be stimulated as part of a sympathetic response, right? The fight or flight, we talked about, your pupils dilate, your heart speeds up. Another thing that happens if you're a guy, your testes get pulled up close to your body, kind of hopefully out of danger. So the cremaster muscle is also about that, kind of pulling them up close if you're having a kind of fight or flight thing, as well as obviously the temperature control. There's going to be a little, what we call the spermatic cord, that kind of goes through that inguinal canal that goes down here. We'll look at this in more detail in a little bit. Right, because the testes are outside of the body, they need this big long cable, basically, that has the blood supply, the nerve supply, as well as a tube to carry the sperm that are getting made. Because the sperm are going to ultimately come out the tip of the penis. So, and that's connected to the urethra and the bladder. So that vas deferens is going to take the sperm and allow them to enter the urethra, so they can get into the tube, where they're eventually going to get ejaculated out. Other big picture things here. I should mention the blood vessels are actually pretty cool. If you look at looking at the time, I'll mention this briefly. So let's draw the testes again. This is the scrotum. If you look at the artery coming down, it's sending the blood down into the testes. The vein that is returning they call it the pampiniform plexus. The veins create this big network that surround that. So what's kind of cool here is as the blood is returning, this is the blood that's already been out, hanging out there, getting a little bit of a chill breeze. So it's kind of cooler already because it's been outside. And now as it's returning back up into the body, it's surrounding and pre-cooling the testicular arterial blood that's coming down. So that's another way you can kind of keep things a little cooler, is by having the returning blood pre-chill the blood that's incoming, which is kind of cool. If we look in the testes in more detail, there's two main things that are happening. One is sperm production. One is going to be testosterone production. So let's look at each of those in turn. So there are all of these little tubules. I think it's something like a half a mile of them in your testes. The seminiferous tubules, these are where sperm is produced. They ultimately all kind of converge in this area called the rete testes. There's then this area called the epididymis, which is basically a place where the sperm are kind of stored and matured. And then there's the vas deferens, which will basically take the sperm up into the pelvic cavity where they can get dumped in mixed with the rest of the stuff that'll make the semen and end up in the urethra to get ejaculated. So the vas deferens is basically how the sperm get up. The sperm are created they're pretty non-motile. Right? It wouldn't make sense for the sperm to be bouncing off the walls all the time. It'd be a big waste of energy. So when we talk about ejaculation, we're gonna talk about things like from the prostate gland that mix in to kind of wake the sperm up, get them swimming and stuff. Like when a sperm is first ejaculated, it actually can't fertilize an egg. It takes a while. There's this process called capacitation where the sperm kind of wake up and become fully viable. So when the sperm are made, they're kind of moving along. There's little peristaltic waves of the muscle in the walls of these tubes that keep them moving along. And if a guy is not ejaculating, if the sperm are just hanging around and hanging around and hanging around, there are cells in the epididymis that can just phagocytize and recycle sperm if they're just lying in the wings too long. So seminiferous tubules. The other cells that we find in here are they're called interstitial cells of Leydig. These are the cells that actually make testosterone. Interstitial means in the spaces between. So they make testosterone. What hormone is going to stimulate them to make testosterone? This is something we talked about on Tuesday. Anybody remember?
Is it the luteinizing hormone?
Exactly. LH. So LH causes ovulation and the creation of the corpus luteum and progesterone in the woman, but in a guy, it just talks to the interstitial cells of Leydig and has them make testosterone. For that reason, if you're just looking at a guy, sometimes they call luteinizing hormone interstitial cell stimulating hormone, ICSH. Those are just synonyms. So interstitial cells. So when we're thinking about the testes, they're doing two independent things. One is the interstitial cells are making testosterone. Then we have these tubes where sperm are getting made. Now we can look at both of these in a little more detail. So I'm going to have to So what hormone from the hypothalamus is going to start this whole process? What triggers the... So interstitial cells are being stimulated by what?
Luteinizing
LH.
hormone?
Yeah, LH. LH talks to the interstitial cells, which then release testosterone. What is triggering the pituitary to release LH?
FSH.
No, that's kind of in parallel.
Gonadotropin releasing hormone.
Exactly. So gonadotropin releasing hormone is being released by the hypothalamus. In a guy, it pulses out every few hours. This is going to be really different than the woman. When we get to the female side, basically gonadotropin releasing hormone is going to be rising and falling on a monthly cycle. But if you're in a guy, it pulses out every few hours. And nobody knows why, but it has to pulse. If you just keep it at an elevated level, the system doesn't work right. So every few hours it pulses, which means LH is getting released, as well as FSH, which will follow in a few moments. The LH then talks to the interstitial cells. The interstitial cells make testosterone. And then we have a negative feedback loop here. The testosterone actually makes the pituitary gland less sensitive to gonadotropin releasing hormone, so it's going to decrease the amount of luteinizing hormone that's being released, right? So we have a negative feedback loop, like we've been talking about. If testosterone's getting higher, there's less luteinizing hormones, so then you decrease the production. When testosterone drops, it releases that inhibition, so LH rises, which means we make more testosterone and it comes back up. So does that make sense? So that is kind of the story with the interstitial cells. On the other side, the other thing in the testes are the seminiferous tubules that are making the sperm. Let me start on another page. Right, so I said if this was a testes, you just have all these little tubules in there. And then if we do a cross section of one of those tubes, basically it's made up of these special cells. They one of their kind of fun names are nurse cells or Sertoli cells. So these cells that go all the way from the outside to the inside. These are my what we call Sertoli cells. And that whole process I was talking about with the spermatogonia, following all those steps till we ended up with actual little sperm. That's spermatogenesis. All that meiosis is going. Those cells are actually being kind of enveloped by the Sertoli cells, and they are being this process is promoted by what we call paracrine factors, things that the Sertoli cells are releasing that keep the process going. So this is spermatogenesis happening, promoted by the Sertoli cells. Ultimately, you end up with the little sperm that are in the lumen here. Sertoli cells, these are listening to FSH. These are stimulated, I should say, this process so basically FSH, which is being released from the right, gonadotropin releasing hormone has two effects. It's going to cause the pituitary to release LH, but it's also going to cause the pituitary to release FSH, right? FSH was the other gonadotropin. That FSH is going to talk to the Sertoli cells. This is going to be my seminiferous tubule here. FSH is going to talk to this, is going to stimulate the production of the little sperm until we get more and more little sperm. If there are enough sperm, there's another hormone that gets released to suppress FSH called inhibin. So this is going to be yet another one of these negative feedback loops. You know, gonadotropin releasing hormones just always pulsing out, which means FSH is coming out. FSH is talking to the Sertoli cells here in my seminiferous tubule, which is then promoting the development of sperm. But if we got enough sperm, inhibin is coming back as a negative signal telling the pituitary don't release as much of this because we got to slow down sperm production. We got enough sperm. So does that make sense? All right, so the last piece we need to add in here then is just how we get these sperm kind of out mixed up with what they need to get mixed up with and then squirted out to where they're going to go. All right, this is supposed to be a guy with a, obviously he has two testes. I just put one here. The word testy is actually kind of fun. Testi, it comes from the Latin to bear witness. Testify, think about to testify. But instead of like putting your hand on the Bible, like in older days, people would like put their hand on their family jewels to swear an oath. So obviously the sperm are getting produced here, but they're going to have to come out here. So we got to look at some more of the plumbing. So obviously the urethra usually is going to go up, and it goes, there's the urinary bladder. Here's the urethra. And that vas deferens is going to go up through that inguinal canal that we talked about and bring the sperm where they can get into the urethra so they can ultimately get out of the penis there. So that's going to be one part of our, when we think about semen, that ejaculate, some of it is going to be from the testes, along, the sperm along with a little bit of fluid, but that's like maybe only 10% of the semen. So if you think about semen, that white goo that the guy ejaculates, maybe only like 10% is coming from the testes. The rest is from these other glands. There's going to be the seminal vesicles. They're on either side. These are going to maybe be 60% of the semen. The seminal vesicles have a variety. One of the things they got is sugar to give these little dudes some energy to swim. Another thing that they got is fibrinogen. Which you've met a couple of times now. What would fibrinogen do? Oh, you all know what fibrinogen does.
Makes fibrin.
Yeah, it turns into that clotting stuff, right? Fibrin. So after the semen is ejaculated, it actually gels. So like when the guy first, sends the semen up of the female reproductive tract, you don't want it just pouring right out. So it goes up and it actually kind of gels, it kind of holds on there, and then kind of reliquefies a little bit later. So the other part of the semen is from the prostate gland. The prostate gland is actually kind of encircling all the way around. It's kind of like, imagine you had a walnut and you drilled a hole through the walnut, and it pours its things into the urethra, but it's actually surrounding all around the outside as well, the prostate gland. It's maybe like 30% of the semen. Ironically, it's named prostate gland because they thought that it was releasing prostaglandins, which actually are not being released by it. They're actually being released by the seminal vesicles, and those like basically help send, like, kind of put contractions of the female reproductive tract to kind of try to throw the sperm upwards. Like the sperm can get thrown quite a bit far up without having to swim, even just because the female reproductive tract is helping them along. Prostate gland, though, does help capacitate the sperm. So like I said, the sperm are developed in this kind of suspended animation. It's not until they mix with this stuff from the prostate gland that they kind of wake up and become really swimmy and capable of fertilizing an egg. So that word capacitate, you should know, basically means to kind of wake up and create the sperm into its kind of viable state where it can fertilize an egg. So when, and I should also mention there's one other gland we can add in here. Let me put them in blue. This is going to be the bulbourethral gland, or the Cowper's gland. This one here, this is not about the semen. This is basically kind of mucus. It's what we, usually what people call precum. Right? When a guy is being aroused, there's this kind of little slippery clear drop that shows up at the tip of the penis. Right? If you think about what is the urethra usually doing, it's like just about peeing and conveying urine and stuff. And now you're about to like send these like sperm up into the female reproductive tract. It'd be nice to kind of clean out the pipes a little before you do that. So this bulbourethral gland makes this kind of mucus stuff that can then kind of clear the way in order to kind of get rid of stuff and get it ready for the reproductive function. And again, it shows up before the guy ejaculates, just during arousal, as that kind of little clear slippery drop at the tip there. If the guy is going to ejaculate, we have to think about the sphincters. Remember the urinary sphincters? What were the two main urinary sphincters?
Is it external and internal?
Exactly. So there was the external sphincter, which was more around the pelvic floor to try to keep the, that's the voluntary one. And then there was one that was the internal sphincter, which was the one that relaxed just due to stretch. So we have these two sphincters. The internal and the external sphincter, and they basically squeeze on either end of what we call the prostatic urethra here, this little bit of urethra where the seminal vesicles, the prostate gland, and the vas deferens all dump into. So when we think about ejaculation, there's two steps. The first step's called the emission phase. So emission phase, that's when all of the ingredients are mixing up. Those two sphincters are shut down. The sperm are getting sent up through the vas deferens. The seminal vesicles and prostate gland are squishing their stuff in and all mixing up to become the semen. This is when the guy knows he's going to come. He can't not come. He can't stop it, but nothing has squirted out the tip of the penis yet. Right? So this is when everything is mixing together. And then the last part will be the expulsion phase. Expulsion phase is now that everything's mixed up in here, this lower sphincter will relax. There are muscles in the pelvic floor that help squirt things out, and it's like spooge. It's all coming out the tip of the penis there. So that's kind of the process there. The last thing we should mention is just obviously, to achieve penetration of the vagina, you can't have the penis be this floppy thing here. So there's penile erection. So let's just talk very briefly about that. If we do like the cross section, like the penis has this spongy tissue in it. Oops. I'm not going to go so much into the detail of it. There's two big rods that go along the shaft, another little one that goes along the urethra, the corpus cavernosum, the corpus spongiosum. So this is what we call spongy tissue. It's called spongy tissue because it has a lot of kind of holes and gaps in it that can fill with blood. So during sexual arousal, we have vasocongestion, meaning filling with blood. This is going to be a commonality in both male and female sexual response. It's more obvious on the dude because you can see the penis getting hard and going up. But in a woman, you actually have the uterus kind of folding back, the vaginal canal elongating, the insides of the thighs becoming more filled with blood. The lubrication in the vaginal canal is resulting from vasocongestion even. So it's more obvious in a guy because you can see this. You see this more obvious thing happening, but vasocongestion we're going to revisit in the female as well. And actually in the clitoris, the head of the clitoris, the glans clitoris, they call the glans of the penis. The head of the penis is the glans penis. Glans, it means acorn actually, because especially if you still got the prepuce, the foreskin on there kind of looks like the cap of an acorn. So glans penis means actually the acorn of the penis, which is kind of hilarious. But in the clitoris also has spongy tissue that is going to swell up during sexual arousal as well. But in the guy, the spongy tissue is going to cause the erection of the penis. We call the crura here, the crura of the, these are called the corpus cavernosa here. These anchor onto the abdominal wall. So when the penis is becoming erect, it's not just kind of dangling there. It's, coming up with respect to the rest of the body. So, trying to think if there's anything else here. So, for the guy, that's kind of it. For the woman, we're going to have a little more that we'll have to cover because there's also the secondary function. The guy is mainly about making gametes and sending them off. Whereas the woman is also going to be about providing a place for the little embryo to develop. So we are going to cover that when we come back after a little break. But we've already covered kind of the core theme of the female side by looking at what's going on in the ovary. What we're going to do is just tie that into what's happening in the uterus and also tie that in also to that bigger picture of the hypothalamus and the pituitary, which is then controlling the other things. And then all that's feeding back. So we'll be looking at that. I should actually mention one last thing. Male sterilization is, vasectomy. Basically, you take the vas deferens and you just cut it. You can actually cut it. You don't even have to go into the pelvic cavity. You can just get it from the outside through the scrotum. And if you cut that, then the sperm don't mix into the rest of the semen. What is the ejaculate going to look like? Are you going to notice anything when the dude ejaculates if he's had a vasectomy?
Yes.
What would you notice?
Well, I just mean, wouldn't there still be ejaculate?
Yeah, so it wouldn't look very different unless you had a microscope and looked underneath and saw there were no sperm. You wouldn't really be able to tell if you had a vasectomy or not, because 90% of that white goo is coming from those other glands. So the only thing missing are the sperm, basically. So it'll still be kind of white goo coming out, just no sperm inside. So, all right.